Tiffany, the floor is yours.
Thank you so much. Thank you everyone who has made time to join us live today and folks who will be listening to this later on. I'm really grateful for the chance to speak with you today about one aspect of the work that I'm doing as the Vice President of Innovation Partnerships at Explore Mars. So today we're going to be talking about one of my favorite things, which is synthetic biology. Synthetic biology is a discipline that merges biology, engineering, and other disciplines to design and construct new biological parts, devices, and even whole systems that don't exist in the natural world. And with synthetic biology, we can also redesign existing biological systems to perform specific tasks. And I am going to try to convince you today that those tasks are going to be ones that are crucial for enabling sustainable human exploration and settlement of Mars in particular, but we can also look beyond not just Mars, but Leo, lunar, and then possibly even further abroad. So slide 2 is where I am now. So usually when I tell folks who are not biologists about synthetic biology, the first thing I ask them to do is to imagine a manufacturing technology that is self-replicating and self-repairing, modular, incredibly flexible in terms of input. It's programmable and reprogrammable with great precision. It's remarkably resilient, and this technology can be stable for years to centuries to millennia as long as you keep it dry and cold. And while that may sound like a really tall order for technology to invent, it turns out we don't have to, because that technology is life. And thanks to 4 billion years of evolution on this planet, we can access this amazing, I'm making air quotes that you can't see, manufacturing technology. So slide 3. As I mentioned, I am part of an organization called Explore Mars, which is a nonprofit that's dedicated to establishing a human presence on Mars, a sustainable human presence on Mars, no later than the 2030s. And as part of that, at Explore Mars, we're deeply interested in the enabling technologies and the innovation spaces that are going to be key for not just getting people to Mars and having them survive there, but also to having people thrive on Mars, whether it's for years or decades or generations to come. And that's why my role as the VP of Innovation Partnerships is so exciting to me. Rockets are amazing, but we need so much more than rockets if we're going to make the human future truly multiplanetary. Slide 4. So more than that, at Explore Mars, we're also particularly excited about innovations that can benefit people both on and off Earth. And that's why I spearheaded a synthetic biology working group that was focusing on the promise of SynBio and all the various technologies underlying it for the space industry. So as I mentioned a few minutes ago, synthetic biology is harnessing and reshaping the natural capabilities of biological organisms in order to address complex challenges across applications. And we'll take a look at a few of those in a few minutes. As part of this working group, we paid special attention to ways that synthetic biology for space could also be synthetic biology for Earth. And so we assembled a team of deep subject matter experts in our group. So Lynn Rothschild from NASA Ames and Brown University joined us. So did Erica De Benedictis from the CRIC. She's also the founder of Pioneer Labs, which is a new organization that I'll be telling you about at the end of my talk. And we were also joined by Niels Averish, who is, I believe, at this very moment at Stanford, but he is moving to a faculty position at the University of Florida as we speak. So we had this great working group, and we also made calls to other folks to get their opinions about what's going on in SynBio for space, what are the big bottlenecks, and what are the opportunities that can really leverage SynBio for space and Earth simultaneously. So at the end of this, and I'm making air quotes again, we have a manuscript that is in review now for publication at New Space. So what I'm going to do today is give you a preview of what you can expect to see in that manuscript, and then a lot of the thinking that went on kind of behind the scenes. Slide 5. And just to tell you a little bit more about me for a second, I'm a molecular biologist by training. My innovation work is and always has been focused on the biotechnologies. It's so great, Penny Boston, to hear your voice on this call. I'm sure Penny doesn't remember, but back when I was a PhD student at Princeton and I was heading out for my first Mars simulation, Penny sent me a very inspirational email that I still remember fondly. So thank you, Penny. I'm still doing it. So as a molecular biologist, the reason that I think it's such an exciting time to be in the biosciences field is that we're at this exciting moment where, thanks to various other technological breakthroughs, the barriers between the living and the non-living world are breaking down in ways that have never been possible before. And the catchphrase that I like to use for this is to say that today biology is technology, and today is the slowest and hardest and dumbest it's ever going to be moving forward. So I personally believe that, electricity was the key innovating, the key innovation and the key enabling technology of the late 19th and early 20th centuries. Computers were that for the late 20th century. And now we're seeing AI and I believe biology fundamentally being as transformative to this century as those technologies were to previous centuries. So it's an amazing, amazing time to be a biologist. So slide 6. Now, I just sold you on this idea that, the 21st century is going to be the biology age. And there's actually a huge amount of opportunity here. So I've seen various reports trying to estimate what kind of global impact we could be talking about. Kind of a median number that I've come across that I believe is a projected global annual impact of 2 to 4 trillion US dollars coming from the bioeconomy by 2040. And so that and other reasons are why the White House issued an executive order in September 2022 that was focusing on advancing the American bioeconomy across a variety of application spaces. You can see them listed here in the screenshot: health, climate change, energy, food security, agriculture, supply chain resilience, and of course, national and economic security. And I would argue also global security for all of these things. So I really like that this executive order also took a step further to say that a bioeconomy, we want a bioeconomy, a thriving and prosperous bioeconomy, but we also want one that's sustainable, safe, and secure. So slide 7, I'm showing you that the USA is not the only country to spot this huge business opportunity in synthetic biology and biomanufacturing these spaces. So just a few months ago, in March of 2024, the EU published a strategy that they called Building the Future with Nature, which, of course, I loved that. And Europe's not alone either. So on the left of this slide 7, I'm showing you a map that came from a review in December 2023, showing countries that had either a formal bioeconomy strategy, some sort of related bioeconomy strategy document, and then the ones that I'm showing you in white here have no bioeconomy strategy that we know of today. So the map that I'm showing you here is from December 2023, and I know it's out of date because, for example, UAE is active in biotech, and Saudi Arabia plans to be a biotech leader based on their Vision 2030 plan. And of course, we know that UAE and Saudi Arabia are also pursuing active space sectors. So my spidey sense is tingling and thinking we're going to see some overlap there. So all of this opportunity around the bioeconomy, synbio, and biomanufacturing will enable us to develop innovative products and processes that can impact the existing processes that we have here on Earth today. That'll help us do things like change the availability of input raw materials, which overall what we're trying to do, of course, is overcome scarcity fundamentally. And I would argue a big reason to do this on Earth is to switch us over as much as possible from our various supply chains and processes that rely on the petrochemical industry. So the potential wins here are to improve the sustainability and the resilience of our ability, our supply chains, and all of these things across these industries. And so that's the story with Earth. If you look at slide 8, I'm showing you that a big goal for the bioeconomy is to improve resilience and sustainability on Earth. And that maps across many of the UN Sustainable Development Goals, which I love. I think they're great. And when I talk about this to business leaders, one of the things that I'm always really careful to point out is that moving to this more resilient and sustainable future isn't just about protecting the environment. It's fundamentally about socioeconomic development and supporting our various industries that we have. So that's kind of like the nice bonus here. We can use biotech to achieve these UN SDGs, but also to lead to prosperity, I believe, for many people in many places around the world. If you haven't had a chance yet, take a look at the great work that the UN is doing on mapping their SDGs to the space sector. It's very interesting, and I think there's a lot more that can be done there. So if you look at slide 9, I'm pointing out to you that it's not just about a circular bioeconomy on Earth, but I see a strong future for a bioeconomy in space over both the short and the long terms, for all the same reasons that I argued for a strong bioeconomy on Earth, those benefits and affordances. So SynBio has several unique advantages with regard to space versus terrestrial applications. So I just want to tell you about a few of those. So first, SynBio has the potential to solve upmass constraints on missions, which would enable us to increase mission capabilities. Secondly, and this is an interesting one, there are no indigenous manufacturing industries or jobs off Earth that would be disrupted by designing systems and processes that are inherently sustainable. So it's not like on Earth where we ask what happens to the coal miners, what happens to the oil frackers. We don't have coal miners and oil frackers on Mars. So the idea of building sustainably from day zero doesn't come with that economic and political baggage that trying to do it on Earth does. And a third big potential benefit in the affordance for SynBio in space is the potential for establishing new jobs and new economies through SynBio in space. And that's attractive for all kinds of things, for investing in space-specific education, functionalities, and markets that tie back to the terrestrial economy and the terrestrial bioeconomy. And last, I just want to point out that I think SynBio is going to be key to addressing the recommendations of the National Academy's 2023 Decadal Survey for the Biological and Physical Sciences. That's the BPS Decadal Survey, which are all aimed at empowering humans to live and travel in space. So slide 10, now I want to tell you a little bit more about how you've probably already seen or heard about or experienced how bio-based systems are being used in space. So the concept of the bioregenerative life support systems, which are known as BLSS or BLIS, are also called closed or controlled ecological life support systems. Those have been explored since the beginnings of the human space exploration era in the 1960s. So a BLSS is made of several interconnected compartments that are based on living biological organisms whose waste of one organism is the vital resources for the other organisms and the other compartments. So you've got biological producers, consumers, and recyclers in these systems. And you could add other things like animals or insects to these loops to serve as protein sources, for example. And also folks have really considered the psychological benefits of having non-human living things in these isolated environments. So there have been several large-scale ground-based demonstrations that have tested closed-loop systems with humans in the loop. So the Biosphere 1, 2, 3, 3M in Russia, Biosphere 2 in the USA, the Closed Ecology Experiment Facility in Japan, and Lunar Palace 1 in China have all integrated these types of bioregenerative life support systems. Other facilities in Antarctica have tested independent functions like gray water recycling at the Concordia station or food production within the Eden ISS mobile test facility at Neumayer Station 3. And then, of course, there have been analog missions, space analog missions that have looked at these bioregenerative life support systems. So Mars 500 in Sirius in Russia, Hera and LML-STP at NASA's JSC, KSC's biomass production chamber, and the MDRS in Utah, where I have been personally, as well as the High Seas facility in Hawaii. So again, like bio is not new news to the space industry. If you look at slide 11, you'll see another example of this. You might have heard of the Melissa system from ESA. So Melissa here stands for Microecological Life Support System Alternative. And it's a project that was started way back in 1987. Melissa is a system of 5 interconnected compartments. I show you a bit of the map here of how these are related to each other. And there's been testing of this system on the ISS for cultivating a cyanobacterium in a photobioreactor in order to convert carbon dioxide into oxygen biomass. And today there's still ongoing ground-based testing at the Melissa pilot plant in Barcelona, where rats are actually serving as the crew. I like that. So if you go to slide 12, I'm going to ask us to go back to this concept of the bioregenerative life support system, and let's focus now, the rest of our time, in this area that I've put in a purple box, which is listed as bioreactors, the microbe compartment. So I want us to spend the rest of our time thinking about microbes, which is just about my favorite form of life on Earth, even though I have a child. Okay, so let's go to slide 13. So as you can see from this map, and hopefully you'll notice in the lower left of my slides, I've given you the citation for these papers. So if you're interested in doing some more exploration on your own, I think these citations to the literature are great places to start. So as you can see from this particular figure, a bioregenerative life support system is just one part of the role that microbes are likely to play in our future in space. So microbial studies in space and other extreme environments here on Earth have shown that bacteria and fungi can adapt to off-Earth conditions. And that's a huge benefit for us because that means we don't have to invent this stuff from scratch, right? Evolution is serving as a tailwind for us. So some of these changes that happen, like biofilm formation, can impact astronaut health and spacecraft integrity in a negative way. We definitely know that. But there are other changes that can promote self-sufficiency and sustainability in space, particularly with humans intervening in some way, for example, at the genetic level. And I'll show you what that can look like. So I would argue that we should be including microbes in our planning and decision-making and mission designs for long-duration missions, not just astrobiome microbiome studies. Let me try that again. Not just astronaut microbiome studies to protect against infections and immune system dysfunction and bone deterioration. We saw some great data just a few weeks ago get published about that. But I'm also talking about biological ISRU studies that incorporate microbes to act as radiation shields or to create electricity and fuel and to make, plant habitats that will enable us to have fresh food and recycling of waste. And most importantly for today, and where I'm pointing with this yellow arrow, is I think we should also really be focusing on how we can use microbes to produce the things that our astronauts will need for long duration missions, for example, like going to Mars. So slide 14, I'm showing you kind of that's the far off vision. If we look a bit closer in, to the potential benefits of SynBio for the exploration phase, specifically, of human activity on Mars. So our team took a look at the 17 NASA technology taxonomy areas from 2020, and we decided to focus on 3 of those taxonomy areas in particular that we think SynBio has particularly strong potential for. So yes, there are non-bio-based solutions at work for these taxonomy areas as well. But we think that bio-based solutions have advantages that non-biotic solutions lack. So, for example, I started this talk by pointing out that biology is self-perpetuating, modular, flexible, programmable, resilient, surprisingly stable. And as I mentioned a minute ago, like there's two more affordances of SynBio for these taxonomy areas that we think are particularly important for space. One is that life has the power to help mitigate many of the mass constraints of space travel by using or reusing resources and basic feedstocks that we have available en route. And that could be like through loop closure, waste, packaging reuse, and or are available to us at the destination. So that's ISRU, and that's things like water, atmospheric carbon and nitrogen, and minerals on Mars. Which means, and here's the headline, instead of trying to store food and medicine for more than 2 years, you can make what you need when you need it. And for us, that's a huge deal in terms of the potential for humans to not just survive these missions, but to thrive during them. So slide 15, I'm showing you an example here from Lynn's lab. So for example, you could use biological sources of materials for off-Earth construction, like mycelium-based biocomposites. Mycelium is the root-like structure of a fungus that has a massive branching filament. Lynn has been investigating using mycelium biocomposites for building in extreme environments on Earth, plus off-Earth environments. So her team has 3D printed a scaffold that they can coat with nutrients and agar, and then that scaffold directs and supports the growth of these fungal filaments. So, and they've also tried using Martian regolith stimulant with just a minimum of addition of biomass or nutrients to act as a binder. And you can see in the upper right of this image, like it worked. They were able to do this. They were enabled to construct inhabitable surface structures. And it looks like from the picture you see here on the bottom, they're envisioning different ways of using this biology and these biocomposites in order to achieve various material properties as well as differential functions based on what part of the structure you're actually working on. So this is just one example of something that's happening today looking at this field. But now what I want to do is go ahead to slide 16 and go back to this idea of harnessing biology as technology. And so an important point that I make when I'm talking to the public about SynBio is that I don't mean synthetic meaning fake. Instead, I'm talking about reshaping and harnessing the natural capabilities of biological organisms with human intervention. So just in case you are not a biologist and you don't spend all day, every day thinking about how biology works, which of course is what happens in my house, just as a quick review, the reason synthetic biology can work is because all life on Earth is related and all life on Earth uses the same information storage system, which is DNA, right? Computers store information as zeros and ones. Life on Earth stores information as the building blocks of DNA, A, C, T, and G. And so DNA is both a coding language and a parts list, if you want to use a computational mental model. And so now, thanks to exponential biotechnologies like DNA sequencing and DNA synthesis, both the coding language and the parts list are effectively open source. So you can take one of these parts out of an organism and move it to another one. That's called genetic engineering. We've known how to do that since the 1970s. You can go in and you can build DNA code from scratch. That's synthetic biology. That's getting cheaper and faster and easier to use. Or you can go in and you can debug the source code of life one letter at a time. That's gene editing. If you've heard of something like CRISPR, then you're familiar with this technology as well. And what's great is because all life is using the same systems, more or less, you can take those parts out and move them among organisms. So you can add parts to a list, meaning the genome of a cell, and the cell will build it for you, and those parts will do things. This is a gross generalization. There are details here that are important, and synthetic biology is both hard and really, really cool. I don't want to undersell that. But the idea that we can think of the genome like the chassis of a car and then program new components to it using DNA is a really, really powerful mental model. The living thing is the hardware and the DNA is the software. So on slide 17, I'm just showing you three basic flavors of how we can use synthetic biology to make the outputs that we want. So biology is incredibly flexible. It gives us a few options. So one option is that we can treat the cell as a living factory filled with machines, DNA and proteins and other molecules that make the things that we want. If we do this in big tanks with live cells, it's called precision fermentation. Picture like a tank that you would make yogurt or beer in. That's what we're talking about. So precision fermentation is a great technology for things like medicine and molecular materials and nutrients. And this is happening today, particularly in the food industry here on Earth. Another method of synthetic biology breaks open the factory, right, the living cell, and keeps the biological machines that are inside the cell, so proteins and other molecules. That's called cell-free synbio, and look for that more in the future. And then a third category is when the whole living cell or a group of living cells is what we're after. So think probiotics, think cell therapies, think living sensors, think lab-grown meat or seafood. That's whole cell synthetic biology. So the key is that in each of these cases, the DNA software has been manipulated by people, right? That's what makes it synthetic. So where does all this DNA code come from? Like, where do these modules come from that we're using as the functional parts of synthetic biology? So take a look at slide 18. More or less, with important details, we can look to all of nature thanks to super cheap DNA sequencing, because there's this huge natural parameter space that evolution has already explored to make cells and to make molecules and parts of cells that do things. So from the bacteria that live in your body to cells that we've been using for thousands of years to make yogurt, like lactobacillus, you can see in the upper left here, to living cells that have important functions like photosynthesis, you can see that in the cyanobacteria. These are all single cells up in the top left of your screen. And there's so much natural diversity here. And you might say like, okay, Tiffany, that's great, but where are we? If you look all the way down in the lower right-hand corner of this figure, which is the map of life on Earth, all fungus and all animals on Earth, including us, are all way down in this little teeny tiny corner. So all of this natural space is fair game for building massive databases of natural DNA sequences that could be used for synbio. Now notice I keep saying natural, but wait, there's more here. If you look on slide 19, I'm pointing out that today with AI, we're gaining access to all of the biomolecules known to science. So in my opinion, the single most important achievement that has been done with AI has been in biology. I am biased. I completely understand. But that huge breakthrough is from these AI models that can predict a protein's structure from its sequence. And the reason we would want to know a protein structure, again, think of the proteins like the machines of the cell, is because the structure of a protein tells you what its function is fundamentally. That's one of the basic concepts in biology. Structure equals function. And it used to be that, you could spend 8 or 10 years or even more crystallizing a protein to figure out what its three-dimensional structure was. Like even when I was in grad school, that was an 8-year project. Now, in July 2022, Google DeepMind released an algorithm called AlphaFold, which can predict a protein's structure from its sequence in 150 milliseconds. So eventually, the database that AlphaFold populated eventually contained predictions for nearly all known proteins, and that's 200 million of them, right? So that was July 2022. In July 2023, a different model was used when researchers combined AI, so it was humans plus AI together, to design and test new, novel, not seen in nature to our knowledge, new biomolecular and cellular structures with fundamentally new functions. I'm showing you that here. And then in May 2024, so just 2 months ago, DeepMind released AlphaFold 3. So given an input list of molecules, AlphaFold 3 generates their joint 3D structures, so how all of these molecules fit together. And it models all known large biomolecules like proteins, DNA, and RNA, plus small molecules, ligands. So that includes things like small molecule drugs. And the AlphaFold 3 can model chemical modifications to these molecules. And these, again, are the molecules that control the healthy functioning of cells, and when you disrupt them can lead to disease. Plus, they're the fundamental inputs to materials and foods and nutrients and things like that. So this work with AI is incredibly empowering for synthetic biology because it helps us accelerate our capacity to do things that nature hasn't tried yet. But remember, these models that I'm showing you on slide 19, these are not LLMs. These are not the large language models that people are talking about all the time. If you go to slide 20, we can see that LLMs are also being applied to biology and to synthetic biology because biology is a language. It's a series of languages. So there are various companies and research groups who are trying to harness the LLM revolution in order to drive biology forward. So just to give you a sense of some rough numbers, the total set of proteins that exist in the human body, we call that the proteome, is estimated to number somewhere between 80,000 and 400,000 proteins. That's in each of our bodies, types of proteins. But the number of proteins that could theoretically exist based on the 26 amino acid building blocks that we have is around 10 to the power of 1,300, right, which is a massive number. Not all of these possible amino acid combinations would result in biologically viable proteins, right? Like not everything works. Evolution has delivered these things for a reason. But evolution has probably only explored a small corner of this possible parameter space. And again, that's only proteins. There are so many other kinds of biological molecules out there. So what I'm trying to say, again, this is the most boring it's ever going to be when it comes to synthetic biology. Because of convergent technologies, there's an opportunity to just blow this space open. So if you take a look at slide 21, I just wanted to highlight briefly, I just wanted to go into the weeds for a minute to tell you a little bit about the actual biology and what we would be doing with our genetic engineering approaches in order to get a cell that we have programmed to make or do something that we want. So some of the approaches that you can see on this slide are agnostic to the target that you're trying to make. It doesn't matter if you're trying to make a food or a structural material or a drug, you'll need to use these. But others of these genetic engineering approaches really depend heavily on what you're making and how it interacts within the native functioning of your cell. That's both the plus and the minus of using a living thing as a factory. The living thing has its own agenda. And so as the biologist, you have to figure out how to get your synbio process playing nicely with what's going on inside the cell for the cell's old agenda. So the design, build, test, learn cycle is commonly used here to construct efficient cell factories that have widespread applications. And other computational tools and databases are really important for guiding precise pathway engineering using the approaches I've got up here. You can also see here I've got some expression elements, so that's listed as expression fine-tuning up above. In biology, these are things like promoters and ribosome binding sites and protein degradation tags that are the details of how a cell makes and controls molecules, things inside it. So Several of these have already been identified and designed in order to fine-tune the expression level of the thing you're trying to make. Protein engineering can also improve the properties of the key enzymes in a pathway. Maybe it takes less energy, or you could do it at a different temperature than the endogenous biological system. We would also want to be able to control metabolic flux more accurately and efficiently. So people are trying to do that with things like synthetic scaffolds, like build a place inside a cell where all of these components come together. Modular engineering, so you can move them around and rewire them. And also just de novo genetic circuits that have been rewired for different control purposes or information flow. All of these things have also been developed for other applications and could be applied, must be applied for space in bio. I mentioned CRISPR before. That's that precise and permanent DNA editing. These CRISPR systems should have wide applications, not only to reconstruct whole genomes, but also in reprogramming the networks of how these various biological components talk to each other. And then just the last one I want to point to briefly here is machine learning. I talked a little bit about AI, but if we broaden our view to a variety of types of algorithms, machine learning is one of the older ones there, and it's been introduced for achieving predictive engineering and for trying to optimize these cell factories. So again, the big takeaway here is there's lots to be done and lots of tools that we already have in our tool belt. So slide 22, I'm showing you an image that I made with the help of Dolly, thinking like, what would synthetic biology on Mars look like, right? And hopefully I've convinced you that SynBio is awesome, not just for Mars, but for Earth too. So you might be asking, great, what's the holdup? So if you look at slide 23, you'll see one of the answers to the holdup has to do with the input availability for a SynBio system, what you bring along or what you have available due to recycling or upcycling of mission products or in situ resources. Each mission class that we're talking about will have a unique set of items that are brought with it, right? Habitat assemblies, furnishings, functional hardware, appliances, scientific equipment, tools that either you bring with you or you sent on ahead. And also, each mission will have different components that are available locally. So that'll tell us things about what feedstocks we can use, what processing pipelines are available for us for in situ manufacturing, either in the ship on the way to where we're going or to our destination. And if you check out this paper of Niels's, you'll see lots of detail in this about that. On slide 24, you see an example from the same paper about how you could think more deeply about what could be possible for a given mission and what we actually might need to know in order to make choices in advance. So Niels considered several mission design scenarios, and he calculated the equivalent systems mass for each of those. So briefly, ESM is a method that allows mass, volume, power, and crew time to be converted into a single metric in kilograms equivalent to predict the up mass requirement, and you're doing that as a proxy for expense. ESM is a standard metric for comparing biomanufacturing systems. Unfortunately, today, it doesn't account for things like risk, sustainability, recyclability, complexity, modularity, reliability, like scalability, all of these things we would want to know. So the bottom line here is that using SynBio in space, the details matter, but it's a solvable problem when we have a focus on the mission architecture, what we take and what we can make. That was slide 24. If we look at slide 25 now, this is just a brief reminder of how microorganisms, these bacteria and fungi, can play pivotal roles in developing technologies for sustainable human exploration of deep space. But, and of course this is a big but, we need mature biotech and bioprocesses for closed loop operations of mission functions. And that could include life support. But what we're really trying to do here is reduce supply chain dependencies for the human presence in space. Microbes need controlled environments in order to achieve their desired functions. So let's think about food and medicine for a second to look at how this dream looks. So slide 26, I'm showing you one of the projects, another project that Lynn is working on, which is an astropharmacy. It's a single set of biological tools and equipment that could be used to manufacture small doses of customizable proteins and peptide-based drugs on demand, nimbly and quickly. That's pretty amazing. Her particular project is focusing on bacillus. Bacillus, you can freeze dry their spores and they're like space resistant. And then the idea would be when they were needed, the crew would wake them up in a bioreactor. And then the cells secrete whatever they've been programmed to make, like peptide drugs, which you then have to purify and use. So short term, you would expect for people to take all these spores with them. But long term, what we would really love is for our astronauts to be able to do their genetic engineering off Earth. Slide 27, you'll see we identified 3 important breakthroughs for Mars synthetic biology that we think will also benefit the bioeconomy on Earth. Number 1 of these is bioreagents that are shelf stable, that you don't need a fridge or a freezer for. For Mars, this will be helpful because there's less upmass and less technological complexity. That would be really beneficial on Earth as well for low resource settings. The second thing, important breakthrough we identified are hardware for on-demand biomanufacturing, including small batch manufacturing that we think for the foreseeable future will be key for space. So that means in space we get better flexibility and resilience, and on Earth we can get like personalized medicine. So the hardware's got to maintain control over pressure, temperature, liquid gas composition. We need to have on-demand sample collection. We would love for this hardware to be able to operate autonomously, at least partially, and have levels of containment that are appropriate for planetary protection. Because, of course, we're talking about moving living things off Earth. And we're also, the third category of breakthrough is robust microorganisms to serve as the chassis and the factories. And we would love for these microbes to be able to survive extreme environments, cold, dry, radiation, and to be able to be functional for loop closure and ISRU. Obviously, way more details here. If you look on slide 28, I go way more into detail about considerations for choosing which microbes to use as the chassis organisms. Based on time, I'm going to skip that for now. But if you are really interested in understanding how do we pick which microbes we use, take a look at the reference that you can see here in the lower left. The good news is biology has given us tons of options to start with. And on slide 29, you can see that our teammate, Dr. Erica De Benedictis, has founded a nonprofit organization in the San Francisco Bay Area called Pioneer Labs, where their goal is to engineer hardy critters, that's what they call them on their website, for biomanufacturing that's ubiquitous, reliable, and green. They're focusing at first on 4 microbes that are their areas of interest. One is a fungus that came from Chernobyl. One is an algae that grows on snow and glaciers and sea ice. Another one grows in the presence of perchlorates, which is very, prevalent in Martian soil, and that's Deinococcus radiodurans. Today, people use it to treat nuclear waste, and also a very slow-growing lichen that is very successful in polar environments and has also been shown to survive 16 days exposed to the vacuum of space. So wouldn't it be great if we could start with these microbes, which already through evolution have some of the functions we would really like, and then tweak them to be more suitable for in-space biomanufacturing through synthetic biology. You probably also have spotted that these organisms might also have potential for terraforming. I'm going to leave that conversation here because I don't want us going down that rabbit hole, but looking on very far timescales, you could see that there could be potential for these microbes for terraforming were we to decide that that's a good idea. So on slide 30, I'm just showing you the technology readiness levels of the underlying tech. I'm not going to go deep into this, but obviously we still need a lot of R&D. And on top of these particular things, we've also got underlying all of this an advanced digital infrastructure for better modeling and simulations. And there's all kinds of open research questions that we can talk about more if you're interested. And on slide 31, I'm showing you a proposed timeline that was developed back by Niels in 2021, thinking about how would a biomanufactory be integrated in the missions upcoming that we know about. Because that development has to be done in concert with planned missions so that we can have the critical opportunities that we need to test the subsystems and the models to evaluate efficiency and TRL. So we can come back to this if you're interested, but the headline is there's a lot to be done in the very near future, but there's also a lot of opportunity. And I think as we see new space growing, we're only going to get more and more opportunities to test and deploy these using commercial opportunities as well. So in slide 32, this is our paper that hopefully will be coming out very shortly, Space Synthetic Biology, a Paradigm for Sustainability on Earth and Beyond. I invite you to keep your eyes open for that. These are these critical breakthroughs that we've talked a little bit about today. And on slide 33, I'm just telling you that, we focused today on a couple of elements of what SynBio could make possible, but there's a much larger opportunity for innovation in SynBio that can provide sustainable solutions for Mars and the Moon and space stations, as well as places here on Earth. And there's just so much we can achieve if we look to use synthetic biology to make living things into factories and computers and sensors and power plants to produce the food, fuel, and fiber that we need in ways that are more efficient and sustainable than what we can manage today. And that will be the case wherever people call home. So slide 34 is just how to get in touch with me, how to stay in touch. I invite you to check out Explore Mars, the nonprofit. We connect people and organizations so that we can push the boundaries of the space ecosystem to welcome stakeholders who are passionate about building a peaceful and prosperous multi-planetary future for decades to come. I'm going to stop talking there so that we have some time for conversation. What do you guys think? What would you like to talk about?
I'm out of breath to think this is terrific. We are a bit to death. Actually, this last slide, this is Harley being the moderator, so I get to ask the first question. And this last slide actually is most of the way there. I was going to ask those of us who are non-specialists, how do we follow more closely what you all are doing at a level that we can understand? And I think you've got some of it here.
I do. Coming and following me on the socials is a great way to do it. I can also offer one other recommendation. There is a book called The Genesis Machine by Andrew Hessel and Amy Webb that takes a look at synthetic biology, how it works, and what could be possible. That's one of the best popular books I've ever read about this, and I highly recommend that. It's called The Genesis Machine.
Great, good. Now, then a more specific question, and again, this is just absolutely terrific stuff. Right near the beginning, you described a couple of, biosphere-like experiments that are going around the country where folks get, and of course, obviously exaggerating here, folks get locked up to see if how long they can live or function well in an environment where I assume most of the elements necessary for life are included there. Which one, I know this requires a judgment on your part, which ones should we be following? Which are the most sophisticated and the ones that possibly most duplicate what life would be, most duplicate the issues that you're trying to make here?
Well, if you want to know, if you want cautionary tales, looking at Biosphere 2 is a great one to look at because they had a lot of challenges with that particular one. I particularly am interested in ones who are looking at these bioreactors, which are essentially like these chambers where everything is controlled. And there are several of these folks, several of these are looking at this. So from the analogs, I admit that I personally don't have a great deal of insight into what's going on in Russia and China, but I would be really curious to find that. Maybe Penny can speak to this. I haven't been to MDRS or FMARS in a while. The last time I was there, we did not have a bioreactor with microbes in it. But that is where I would be focusing my attention. Like, where are people testing precision fermentation for space? That's where I would be looking. And I don't have the answer to that off the top of my head, but it's something we can find out.
Okay, good. Great. Thank you. All right. Are there other questions, folks?
Yeah, this is Penny.
Hi, Penny.
Hi, Tiffany. Lovely talk, dear. Thank you for doing this for FISO. I have many things. We should actually tag up and have a conversation. We haven't spoken in quite a long time, but you've laid out a wonderful sweep and an overview across synthetic biology, and it's a super exciting field. I have concerns, and you probably do too, but I want to raise them here. And one is that if we only take one of the possible perspectives on SynBio, we view life as an engineering process, which is very useful, and that's the way that SynBio is going so far for most of the things that I see. But the other aspect to SynBio that I think is equally, if not perhaps even more important, is the notion that we have to use those capabilities to create ecosystems for our built systems. And the ecosystem functioning is a whole set of levels of complexity above the individual components that we can actually engineer. So genetic engineering and CRISPR and all of these tinkering with the guts of the cells are wonderful, but what you're really looking at is the whole system. And so without the ecosystem component of it, which we may not be wise enough to design from scratch, even with AI, because of our fundamental lack of understanding of the basic mathematics of complex systems. You see what I'm getting at? So
I do.
I think we have a lot more work to do, and if we simply approach it as an engineering project using a whole set of new fabulous GUI and biochemically complex tools, we're going to get ourselves in trouble and we're not going to progress. So I just want to, bring that up and hear your thinking about that whole area.
I'm so glad, Penny. Thank you for bringing that up. Yes, so that's both the joy and the challenge of working with biology, right?
Yeah.
Because there's always something else going on in a biological system than what we realize. What we think the system is optimizing for is not necessarily what the biology has optimized for. And like I said, that's like the joy and the terror of being a biologist. If you look on slide 30, which I skipped through really quickly when I was talking about the TRLs and the various components and, the progress that we need to see made, you'll see underneath, I've got this just throwaway text box that talks about computational biology, databases, and simulation. And I think to your point about how are we going to get our handle around the complexity, the dynamics and the complexities of biological systems, especially in cases where there are emergent properties when we move between levels of biological hierarchy, meaning between molecules to cells, cells to tissues, tissues to organisms to ecosystems. There's just so much there that I think we're going to need a lot of help with modeling and simulation. So, the good news / terrifying news is, as you know, biology is a system that is self-correcting, right? Like you will find out what works and what doesn't work. The key, and what I would argue here, to your point, is that I don't think we can afford to wait before we know everything in order to do anything. So it's that gray area that we're in right now that is why I think there's so much potential here for people to solve some of these really big problems, including the ones you're talking about, Penny, which would be hugely helpful for using SynBio on Earth as well.
Yeah, and we're not working the SynBio from that end. Not doing it in NASA, as far as I know, and I don't think we're doing it anywhere.
Huge opportunity.
Yeah. So there's a whole missing piece where we mix the ongoing research that you just described so marvelously, and what we know about deep ecology on Earth, which doesn't, those communities of people don't often stray into the same terrain, and we need to start encouraging them to do that and recruit people from that world into the symbio and space arenas if we are not going to be the victims of our own hubris.
I completely agree.
I don't think we need to wait on anything. We need to start fixing it up.
I completely agree.
Yeah. Thanks for your thoughts. Thanks for the.
Okay. Penny and Tiffany, thank you. We've got time for a couple more questions. This is really fascinating stuff. I've got a quick question. What is the funding source for most of this work, Tiffany?
For space or for Earth in bio?
Both.
Both. So the headline, of course, is that there's not nearly enough funding for this going on. Oh, good. I'm glad other people think that's funny. Right. So, obviously, national space agencies are very important here because when you're talking about space applications for SynBio, there's not exactly a large addressable market for that yet, right, from a commercial perspective. So for a lot of this, we believe that still national space agencies and our basic science funders like NSF, DOE are still super, super important for this. And one of the reasons when I go around talking about this, I really hammer on the sustainability piece is because I believe that solving for the climate challenge, solving for sustainability is also solving for space. So I'm trying to shake all the trees to see what money will fall out, to see what we can find. Now, there are also, though, interesting opportunities in the commercial sector. So I am seeing startups who are starting to do this. The really clever ones, though, are figuring out, okay, we have this product that we want to get out for the space market. What is the Terran version of that from now for the next 10 years or whatever it is, right? Because there's just not enough money-making opportunity yet for the capitalist markets to really drive it. What I think is that maybe, and again, I'll give you a number and we know it's going to be wrong. In Tiffany's Magic Fantasyland, within 20 years, there's enough of a space economy that these things are generating their own investment. And then there are space-focused VCs out there who have investment theses, which are really quite different from folks who invest in apps. They're able to take the longer view. And then one other funding source that comes to mind, of course, is defense. What are the ways that defense could be leveraging these various pieces? There's a lot of opportunity there as well.
Okay, great. This is such great stuff. Why don't we take one? We're a little bit after the hour. Why don't we take one more quick question? And Tiffany, once you get it, try and answer it quickly. So who's got a quick question?
Well, this is Dan. This is probably not a quick question, but I'm going to ask it anyway. It was really interesting. You're talking about the supply chain that's required to do SynBio, say in a place like Mars. And I'm just wondering that putting together organics, in particular, how do you build biological systems if you need carbon? And you don't have carbon in the form of organics. You have it in CO2, I guess. You have it in carbonates. How do you make organics out of rocks and CO2 in say, a bioreactor?
Yeah, so slide 28, I skipped that, but it turns out biology is really very good at fixing carbon. So you've got types of organisms that can do this, like phototrophs use light as their primary energy source for metabolism, and then they drive photosynthesis in order to fix carbon. Natural sunlight on Mars is weaker than Earth sunlight, so you probably need like focused light or artificial light, which is of course more up mass. Or you need to re-engineer the microorganisms to be able to use less light in order to fix carbon. So although there's some organic molecules in the Martian regolith, we think that the gas in the atmosphere, the CO2 and the N2, is probably the most accessible source of carbon and nitrogen for synthetic biology.
Yeah.
So yeah, we probably use a mixture of biotic and abiotic roots to do that. You can see that at the figure on the top of slide 28.
No, actually, slide 28 looks exactly like what I was interested in. I think you did actually refer to the atmosphere as an in situ resource. And on Mars, that's exactly what you'd be doing. Okay, so thank you very much.
Okay, great, Dan. Thank you. And Tiffany, thank you so much.